Okay, so hello everybody. I hope you guys enjoyed the long weekend. Had a nice Valentine's Day last week. I realized I didn't say anything on Thursday, but now we're back to our content. I wanted to start today actually with a quick little apology for last week. I feel like I was starting to get a bit agitated during the Unit 2 review, and maybe none of you felt that, and hopefully nobody did, but basically a couple minutes after we got off Zoom, I realized that I was feeling frustrated and annoyed, and that probably came across. And so I wanted to apologize if it did, and talk about where that was coming from. I certainly was feeling frustrated that there weren't very many questions or topics to review during our class time. And so I think my main frustration was that we weren't utilizing that class time efficiently. Like it felt like a bit of a waste. And it is a small part of responsibility on your end. If you voted to have this review during class time, right, we changed what class was going to be so that we could review, then the expectation is there that you come with questions and topics that we're spending that time wisely. However, a majority of that responsibility is on me as the teacher leading this discussion to have set up this environment in a way that ensured that we utilized that class time efficiently. So again, if you could tell that I was a bit agitated and frustrated, I do apologize for that. That wasn't fair and is unprofessional. Just wanted to start off with that. And wanted to give another opportunity to potentially spend some time reviewing stuff today before we jump into Unit 3 content. So I created a quick poll, which is basically what I should have done for last Thursday. And so maybe there's nothing that you guys want to review and everyone's feeling confident, but I do want to send this out in case anyone is. So the first question is just asking for you to rate your overall confidence with Unit 2 content. Please respond to that one. And then the other few questions are asking you to select up to 2 topics from each chapter that you would want to spend some time reviewing. And so if we have a decent amount of responses here with topics that you guys would like to spend time on, I'm happy to dedicate the beginning half of class today to that review. And then we can move on to chapter 12 afterwards. So please take a minute to respond to the poll. Are you guys seeing that? Okay, yeah, getting some responses now. Still waiting on a couple responses to come in. Okay. I think I have responses from everyone that's here. So I can share the results with you guys. So confidence is in the 2 to 4 range, mostly in the 3s. So definitely want to do what we can to bump that up, get as many people into that 3 or above before the exam tomorrow. So in skin, we'll definitely look at blood supply and innervation. I'm not looking for a whole lot of knowledge here as far as what you'll be assessed on, but we can touch that. And then we can touch on layers, a bit about hair, and then glands I know are a bit difficult. For bone, we can talk about our 2 types of building bone or both types of ossification, and look at the structure of bones and calcium, excuse me. In joints, we can look at synovial joint structure and types, and then for intervertebral discs, and then all of the basically things involved with muscle contraction here, and then energy. Okay. So I will address these topics on my end relatively quickly and then open up for questions if you have specific questions about those things. And I'll try to explain like basically what kind of knowledge I'm looking for in each of these topics so that we would still have some time to start chapter 12. Okay, so let me move that over. Thank you guys for your responses. And let's start with skin. So I'm looking at innervation and blood supply. Basically, as far as innervation, or let me actually start here where I have it. So no epithelial tissue has blood supply directly to it. Right? There are no veins, capillaries, or nerves going attaching directly to epidermal tissue. It is getting most of that information from diffusion. So the nervous side of that, the innervation of our epidermis, is coming from these specialized cells. Where do I have that described? I guess they're kind of in here. I could have swore I had a slide on this. Is it really just mentioned here? I was looking for a slide that showed Merkel cells, and I thought I had a better image of that, but I apologize that I do not. Let's go back here. So in our epithelia, in the most basal layer, the deepest layer of the epidermis, we have these Merkel cells, which are going to transmit the sensations that we are feeling through our skin to a nerve so that we can translate that signal into perception. So as far as responding to sensation, those are Merkel cells in the epidermis. And then we do have nerves and blood supply in our dermis under that epidermis. So under the epithelium with all this connective tissue. And the papillary layer, we have those papilla, which is what gives it the name, these like folds, this little mountainous looking thing. And each of those papillae have the blood supply and the nerves that are going to supply and respond to that epithelia. So all of the nutrients that the epidermis needs are coming from the blood supply in these papillae. Those Merkel cells are transmitting those sensations to the nerves that are pushed into the papillae here. We have a few different plexi as far as where these things are in highest concentrations with the circulatory system in our nerve cells. So we have our subpapillary plexus, which is just under the papillary layer, as the name suggests, subpapillary. And then we have our cutaneous plexus, which is between the reticular layer and our hypodermis. This is just where the largest concentration of those blood vessels are, and then they branch out into the different structures here. So Merkel cells for transmitting signals to the nerves, the papillae providing those blood vessels close to the epidermis so that they can get their nutrients through diffusion. And then we have our two plexi, or subpapillary plexus in between the papillary layer and reticular layer, and the cutaneous plexus just deep to the reticular layer, which is where we have those blood vessels in highest concentrations. So is there anything I could expand on that topic of blood supply and innervation? Okay. Quickly look at keratinocytes and layers of the epidermis. So for layers of the epidermis, it's just going to come down to, can you come up with some memorization technique that makes this stick in a way? I already forgot the one that I shared that comes from Crash Course A&P. I was never great at keeping these memorized, but basically the most superficial is the corneum, followed by the lucidum, which you only find in thick skin. It's a very glassy, clear looking layer of epidermis here. Then the granulosum, which would be the second most superficial layer in our thin skin. Again, that lucidum is only in thick skin. Followed by the spinosum, and then lastly, the basale or basal layer, which is what is going to have that basement membrane attaching it to the papillary layer of the dermis. And essentially, what we have is just different layers that show different stages of where that keratinocyte is in its growth. So we have our stem cells and those Merkel cells as well as melanocytes, which are making the melanin pigments that give our skin its color. Those are all in our stratum basale in this basement layer. I guess that'd be down here. Those stem cells are going to start producing keratinocytes or early versions of keratinocytes, which are going to be our finished product skin cells that will be following all the way up here. Those keratinocytes, as the name suggests, are specialized to produce a whole bunch of keratin, which is a fibrous protein. And keratin is what makes up our nails, our hair, and then, of course, this is what makes up a majority of the mass of our epithelium, or epidermis, sorry. So as those keratinocytes move up and like towards the surface of our skin, so as they move superficially and transmit from layer to layer, they're essentially producing more and more keratin and getting more and more flat as they lose their cytoplasm until eventually in our stratum corneum, they're mostly just dead attached keratinocytes that are now full of keratin. So this largest portion, the largest layer of our epidermis here, these are all essentially dead skin cells, just full of that keratin, which makes them very biologically inexpensive. So if we lose them, that's fine, but they were expensive to produce. Right? So we're not really losing a lot by rubbing off a few layers here or a bunch of cells by rubbing that off, which provides us some protection, etc. But yeah, it's basically all the stuff that we need to sense and produce these cells is down in that base layer, the basale. And then as they move towards the surface, they're producing more and more keratin and flattening until eventually they are completely flat cells interlocked with each other, just full of that keratin protein. No more cytoplasm, no more nucleus, endoplasmic reticulum, etc. They're just there to be keratin. If you have a question on that, you can go ahead and put that in the chat. I'm going to move on to hair follicles and glands. So I briefly addressed this on Thursday, I believe. When the question came up, there's not a whole lot that I'm looking for here as far as you're not going to get a diagram asking you to label like the matrix, medulla, cortex, cuticle, etc. I'd rather you understand the concept of that each follicle is, again, like an invagination of that epidermis. Whoops, didn't mean to move that. So rather than this epidermis fully connecting here, it has been pushed into the dermis, which provides this sheath, this follicle that completely surrounds our growing hair. And so in a similar way, at the roots, which is our matrix here, we have in the bulb, we have most of our stem cells. This is where we have our innervation and reflexes or sensation, sorry, which is going to connect to the nerves from this papilla. As these stem cells are producing more and more keratinocytes, we have melanocytes down here too to produce melanin, again, the pigmentation that gives our hair color, skin color, nails color. As they move through these layers, they are going to become basically flatter cells and eventually will die and be just fully full of keratin, which is what makes the hair that we recognize on the external portion of our bodies. So the only living cells are deep in this, in our root here, deep into this follicle. By the time it is extending beyond the surface of our epidermis, that is all dead keratinocytes full of keratin and melanin, if it has some pigmentation to it. Each follicle, or a lot, I don't think all of our hair follicles, but most of our hair follicles have their own erector pilae muscle, which is a smooth muscle. We don't have control over that. It is automatic. A lot of them have sebaceous glands, which secrete oil directly onto the hair, but we do have some sebaceous glands which secrete oil directly onto our skin. And that's basically all I'm looking for with hair. You're not going to see this image. So just recognizing that general concept, knowing what an erector pilae muscle is, and that some sebaceous glands secrete directly onto it. Apocrine glands are going to secrete onto these hair a more milky, protein-rich version of sweat in very specific areas, our anogenital regions. And that brings us to gland types. Okay. So the two major types we have are sebaceous versus apocrine, or sebaceous versus apocrine and eccrine. And eccrine is also called merocrine. But essentially, you have oil glands and you have sweat glands that secrete onto skin and hair. Sebaceous, it produces sebum, which is what gives it the name, which is a type of oil. This is there to lubricate our skin, helps our skin retain its water, also prevents excess water trying to diffuse into the skin, so we don't end up swelling our skin cells. And then our apocrine and eccrine glands are two different versions of sweat glands. So the eccrine are much more common. These secrete onto the surface of our skin, secreting that sweat that we think of, mostly water with some salt in it. But then apocrine glands secrete basically modified versions of that sweat. These only become active after puberty. Okay? And so a majority of our apocrine glands are secreting that milky protein-rich sweat that I was describing, which would be secreted directly onto our hair and our armpits and anogenital regions. And this is the type of sweat that ends up producing a smell or body odor. Basically, we have a bunch of bacteria living on our skin, and those bacteria are going to eat up the protein that is in that protein-rich milky sweat. And the byproducts that they release are gaseous and have a smell to them. So body odor is that reaction of the bacteria on your skin metabolizing that protein-rich sweat in your armpits and in your anogenital region. Evolutionarily, this would be for recognition of people from the same group or family. It also signals maturity, so ready for sexual reproduction. And then we have a few different examples of modified versions of these apocrine glands. So these are the glands that produce our earwax, that help protect our eyelids, and then produce milk, lactation. Those mammary glands are modified versions of apocrine glands. So those are ceruminous for earwax. Cerumen is the actual name for earwax. We have ciliary glands on our eyelids, and then mammary glands, which produce the milk in mammals. Any questions about the last few topics we just discussed? Questions about keratinocytes and layers of the epidermis, hair follicles and hair, or types of glands that I did not address? Okay, then we will move on to bone. So you guys asked to look at compact versus spongy bone, structure of bones in general, or I think we have more specifics there. And then our two different types of ossification, so creating bone, and then the relation to calcium balance and the hormones that control that. So, see, where do I want to start here? Let's just look at our two major bone types. So in our two major bone types, which are our long bones and flat bones, we've introduced the various classifications, but the most common are those long and flat. We basically have compact bone creating the outer portion. So it's more compact, more structurally stable, providing kind of the, what's the right word? The model that will hold together that bone shape. And then spongy bone filling some of the space in between. So in flat bones, that looks very much like a spongy bone sandwich. We have compact bone going around the entire perimeter and spongy bone filling all of the inner portions, the deep portions. In our long bones, we really only see spongy bone in each of our epiphyses, so the two ends of the long bone. In the diaphysis, most of that space is left empty to be full of marrow and to provide space for the blood supply and nerves that are going to supply that bone. And so that is called our marrow cavity, since it's very full of marrow, but we do have marrow in between the trabeculae of our spongy bone as well. So difference in the structure between compact and spongy bone. Compact bone has these osteons that we talked about, these different layers of lamellae surrounding the blood vessels and nerves that are going to supply that bone. And then we have osteocytes, basically stuck into their lacunae, these osteons stuck in their little lacunae, which is where you see these little pink things sticking out. So we have layers called lamellae circling each other. with osteons stuck in between in these little lacunae. And those osteons are continuously producing more hydroxyapatite crystals and collagen fiber to help upkeep the density of that bone and the size of that bone. Those lamellae are also really important for giving support, resisting tensile forces. Right? So each lamellae has collagen fibers in this parallel orientation, similar to what we saw with the dermal lines in skin. And so they alternate in what direction they are facing. So right here we have collagen fibers going from top left to bottom right. For the next lamellae, the next layer, we have collagen fibers from top right to bottom left. So they're alternating this crisscross pattern, which allows it to resist that like twisting and pulling to give our bones that resistance and strength, allowing them to keep their shape without just breaking, because the hydroxyapatite crystals are brittle. And it's salts holding that all together. So then for spongy bone, the main difference is just that they have these trabeculae. So they don't have lamellae. We don't have this circular formation. I believe I've been saying osteons. They are osteocytes. The osteon is describing this entire circle. Yeah, osteocyte. Sorry, I confused myself earlier. So an osteon is one full unit here, this whole circle. The actual bone cells that are stuck into those lacunae are osteocytes. I said that correctly the first time and then backed out of it. So again, osteons are these entire unit, multiple layers of lamellae, and then osteocytes are the bone cells that are stuck in those lacunae, producing hydroxyapatite crystals and collagen. We see the same general structure just without those lamellae, without those circles. So we don't have osteons, we don't have lamellae, we just have more osteocytes stuck into lacunae in these different branches called lamellae. And so rather than resisting that tensile twisting type force, the spongy bone is going to be better for resisting that pressure from top to bottom and for getting here we go. That body weight force is going to help with that compression with those trabeculae and the hydroxyapatite crystals in general. Help with compression force. The lamellae, which are, again, are those layers, those circles, which have collagen fibers going in different directions, that resists that shifting and twisting type of tension. Compact versus spongy bone, structure of bone. Okay. So now let's look at how we make more bone, our ossification. Oops, that might have messed stuff up. Let's go back. Okay, so we talked about two different types of ossification. And ossification is referring to building new bone. So we have endochondral ossification, as the term suggests, chondral referring to chondrocytes or collagen, and then endo meaning within. So building bone from within our cartilage. And then we have intramembranous ossification. So within the membrane, this type of bone building is going to come from our dermis. So starting from the deepest layer of the dermis to build bone. And this one is starting with a cartilage model and building that bone from within. So endochondral ossification typically creates long bones. Intramembranous ossification forms dermal bones, a type of flat bone. And so how do these work? All right. So endochondral, again, referring to within a cartilage model. So long bones start out as full of cartilage. Cartilage makes the general shape that that bone is going to end up fulfilling. The blood supply that is going to supply our bone penetrates into that cartilage model into the center, which is going to become the diaphysis. And then osteocytes start to form and start producing those hydroxyapatite crystals and collagen fiber. So starting to form bone within that cartilage. We have osteoclasts breaking down bone, and osteoblasts building new bone as this continues to build. So essentially, as one layer is made, osteoblasts are making another layer outside of it, and osteoclasts are breaking down the inner layer. So over time, this keeps expanding and expanding and expanding until you are left with bone surrounding this entire diaphysis and just space in between and the blood vessels that are supplying it. So that space is going to become the marrow cavity that we saw in our long bone. You can see here all this space in between. And then at this point, we're just left with two areas that are full of cartilage still. So that same blood supply that was supplying the building of this diaphysis is going to penetrate into both of our epiphyses with some help from other blood supplies, also going to penetrate in here, and we have the same thing occurring again. So bone is going to build and bone is going to break down as it is replacing that cartilage and expanding until eventually all of the cartilage has been broken down and bone has completely replaced it. So now we have two epiphyses that are fully surrounded by compact bone. We have a diaphysis that is fully surrounded by compact bone. And typically what gets left behind is this epiphyseal cartilage, which makes the epiphyseal plate or growth plate, which is going to break down over time as we age and mature and develop. So that's these empty spots that look like this bone is broken or just completely disconnected. If you look at the x-ray of a child's arm here or hand here, and then an adult's hand, you can see those bones fully connect at their joints. Really, all that space is just the cartilage that was still there in these long bones. That is the general process of building bone this way making sense? You guys have questions that I can clarify? And your biggest hint is in the name, endochondral. So within cartilage, this is building bone with a cartilage model. Just going to make these long bones.
Professor? Yeah.
Go ahead.
Yeah. In the study guide, it says describe how long bone grow longer at epiphyseal growth plate. So it says give the detail of each step. So for each step, it's just what you are saying, right? So we wrote down all the details for each step, or just a general concept, a general ideas about this part.
So yeah, in a question like that, if I was asking you to describe endochondral ossification, the concept part, like the way I've been talking about it, is what I'm going to be looking for. Of course, the details are great and shows a deeper understanding, but typically studying for those details means that you're cramming factual information rather than focusing on building the conceptual understanding. And I'm looking for the concepts, so the basic pattern of the basic steps that I talked about, rather than specific details.
Got it. Thank you. Of
course. Okay. Okay, so comparing that with intramembranous ossification, this is starting from the dermis, so the skin. Instead of starting with a cartilage model, we're basically building the bone completely from scratch without any model to fill. And that, I guess we should go have these. Basically, we start to build those stem cells, these mesenchymal cells, that are going to generate little pockets that are going to become bone. So we'll start ossifying in the same way that we see with any kind of bone building. Osteoblasts helping to make more, and then they end up maturing into osteocytes that are surrounded by the hydroxyapatite crystals and the collagen that they're producing, until eventually we have a bunch of osteocytes stuck in their little lacunae, and they are surrounded by those hydroxyapatite crystals and collagen fibers, which are what make up a majority of the mass of the bone. Okay. And so since we have that separation, and as they expand, they start to connect, that's kind of what makes those these little pockets and ends up forming into those trabeculae that we see with spongy bone in here. We get those little pockets because of the blood supply and the marrow that's going to end up filling those spaces. And it's much less uniform than filling a predetermined model like what we saw with endochondral ossification. So intramembranous, again, is starting from the dermis and the skin. We're getting these little pockets of mesenchymal cells developing into osteoblasts, which develop into osteocytes. And those osteoblasts and osteocytes are going to continuously make more hydroxyapatite crystals and more collagen, which is going to expand the space of these little pockets until eventually they connect with each other. And then that entire space is full of mostly bone. And the only spaces left behind are going to be full of, not cartilage. Why is cartilage coming to mind? They're going to be full of marrow and the blood supply that's going to supply these bones.
So osteoclasts are not involved in intramembranous ossification?
That's a good point. I don't think they are because we don't have the same build and breakdown to expand like we saw with our long bones. I don't think at any stage in here we have the breakdown of bone for intramembranous. So I think I'm safe in saying no, but it's a hesitant no. As far as what you would be assessed on, definitely not. There are no osteoclasts involved here from my end. Okay. Oh, and Constance, you asked a question last class related to muscle, or I guess I can wait until we get there, about how calcium is detached from troponin and reuptake. And I did get somewhat of an answer, not a super clear answer, but at least something that makes it make more sense. We'll talk about that when we get there. Okay, so the last topic from bone was calcium balance and hormones. So this basically burns down to the fact that a majority of the mass of our bone is from those hydroxyapatite crystals, which are basically just ions. And they're formed into these salts that take a structure that provides the support for the bone. And those crystals are mainly made up of calcium, potassium, carbonate, phosphate, magnesium, and sodium. Mostly calcium and phosphate, making up a majority of that mass there, followed by carbonate, and then the rest of these are in pretty small concentrations. But if you look at how much of those ions we have in our bone compared to the rest of our body, you can see that a majority of the calcium, phosphate, carbonate, magnesium are stored in our bone. And in that way, they're not only providing the function of supporting our body as bone, but we can also access them as storage when we need access to more calcium or need access to more phosphate. So we can activate our osteoclasts to start breaking down those hydroxyapatite crystals, releasing these ions into the bloodstream so that other parts of the body, other tissues have access to them. And so, of course, with any mechanism like that, we have hormones that are going to signal when to break down and when to inhibit those osteoclasts, or when to activate osteoblasts to build more, or inhibit those osteoblasts that we stop building more. Basically, if you're going to build more bone, if the osteoblasts are active, we are removing calcium, phosphate, magnesium from our bloodstream. And if we need to access more, we want to activate our osteoclasts, which will break down our bone, thus giving more calcium, phosphate, magnesium to our blood. So it's a balance. And vitamin D plays a big role in that balance with modifying or regulating osteoblast and osteoclast activity. And then we have these hormones that we talked about. And so I included these little arrows to show their general effect on blood calcium levels. So calcitriol and parathyroid hormone are going to be related to getting more calcium into the blood, thus less in our bone. And then the rest of these, growth hormone, thyroxine, sex hormones, and calcitonin, are all going to promote growth of the body or growth of that bone. And so that's going to be taking calcium out of the blood, essentially. And I think the main ones we look at are calcitriol, parathyroid hormone, and calcitonin. So if you can recognize the effects described in basically these two figures, you're probably all right. So factors that increase blood calcium level. So the main ones here, we're looking at parathyroid hormone and calcitriol, with calcitriol basically working in tandem with parathyroid hormone, it's PTH. So parathyroid hormone is going to be the main one that is increasing the amount of blood or the amount of calcium in our blood. And that can either come from our digestive system, increasing absorption or helping activate the proteins involved with absorbing calcium from our food. So from the lumen of our small intestine into the small intestinal cells and then out into circulation. Parathyroid hormone is going to inhibit the kidneys from removing calcium from our blood, from filtering them. And it is going to stimulate osteoclasts to start breaking down that bone. So we have an effect in 3 very different areas, all leading to increased calcium in our blood, breaking down our bone, getting it from our digestive system if there's food there that can provide that calcium, and then keeping it in our blood rather than filtering it out. And parathyroid hormone and calcitriol are both going to be stimulated to be released if our general calcium ion concentrations get below 8.5 milligrams per deciliter. So recognizing that general range, like you saw in the quizzes, could be good information to have. And then the higher end, which is going to stimulate our hormones involved in decreasing the amount of calcium in our blood, is 11. So basically the range of 8.5 being the low end to 11 being the high end is the general amount of calcium concentration that we want in our blood. Anything outside of that range is going to stimulate these hormones to be released as a response to regulate the amount of calcium in our blood. And so rather than increasing the amount of parathyroid hormone and calcitriol, if we have too much calcium in our blood, those levels are going to decrease, or we won't be releasing them at all, so that we will stop absorbing calcium from our small intestines, so that we're not getting more into our blood. Calcitonin is going to do the opposite of what we saw with parathyroid hormone. So it is going to stimulate the kidneys to continue filtering out calcium and secreting it in our urine, excreting it in our urine. It's going to inhibit osteoclasts so that osteoblasts can continue doing their job of taking calcium from the blood and putting that into hydroxyapatite crystals in our bone. And so those are our 3 mechanisms: either increasing or decreasing absorption from our intestines, filtering out or stopping the filtering of calcium from our kidneys, and then either allowing osteoclasts to break down our bone or inhibiting those osteoclasts so that they are no longer breaking down our bone. And those are mainly controlled by parathyroid hormone and calcitriol, which are going to put more calcium into our blood, or calcitonin, which is going to take the calcium out of our blood or help stimulate removing calcium from the blood. Is there anything I can expand upon there? Realizing this is eating up a bit more time than I thought, but I guess this is probably more important right now than starting on the nervous system. We basically have 4 chapters talking about all the various parts of the nervous system. So articulations. You guys want me to look at the structure of synovial joints and types of synovial joints. So it looks like you're feeling good about movements and general joint classification, and then structure of intervertebral discs and ligaments. Okay, cool. I already have our structure pulled up. So our synovial joints, again, they get their name because they're full of that synovial fluid, which provides for more space between the 2 bones that are articulating there, which allows for more movement. So our synovial joints are our most mobile joints. They are diarthroses. And so they have synovial fluid filling them, which come from our synovial membranes. We have menisci or meniscus, which are little pads of either cartilage and protein fibers, or and I also have a mix of fat in there, which tends to make these little triangular looking shapes at either end of the articulation to provide some protection as those joints move and potentially the bones come into close contact with each other. It just provides some of that extra insulation and padding. We have fat pads, which, as the name suggests, literally provides a pad of fat to help with our meniscus. So this is just adipose tissue, again, making a similar kind of triangular shape in between the corners of these long bones, which might come into close contact as these bones are moving at the joints. We have ligaments, of course, helping these bones stay connected to each other, as well as connecting to some of the various structures around it. And then we have these bursae, which are these light green pads that we have all around, shown all around here, which are also filled with fluid, surrounded by synovial membrane, getting more synovial fluid to fill them, which just kind of adds for extra space and mobility. I believe these are also recruited if the synovial membrane is having a hard time for adding more synovial fluid, or if synovial fluid is leaking for any reason, those bursae have the ability to be recruited to release some more synovial fluid and diffuse it into that membrane, which will be diffused into the space. That's generally it. I guess also our articular cartilage. In almost all of our joints, we have cartilage in between the bones to provide some extra there. But really the main difference is that fluid, the synovial fluid, providing that extra space. And then all these other accessory structures are just to provide extra protection and support since there is so much movement at those joints. Are there any specific questions that came up as you guys were studying and reviewing that I could try to address or clarify? Or is this just a intimidating structure since there's so many terms of you? So our types of synovial joints. Trying to think of how to best address this and what kinds of information or insight into the assessment I can provide that isn't too direct. Really, if you can recognize this kind of image showing the planes of movement and connect that with either being monoaxial, multi-axial, non-axial, biaxial, or triaxial. So you can recognize these general images and the name that goes along with them, and then have at least one example of a type of joint that is related to that, you'll probably be okay for any question about this on the exam. Just showing the different ways that bones will move at these various types of synovial joints. So our first example was the gliding joint, like where our clavicle articulates with the manubrium, which allows for some shifting, but is fairly stable compared to our other types of synovial joints. So as the name suggests, they kind of glide along the same plane where they meet each other. So that's where it gets that multi-axial. So we have two axes, our X and Y plane. We have monaxial joints like our hinge and pivot. So like I said, just having one example, like if you just remember the ones that are in these images too, that's probably fine. So like our elbow joint here, so it has this very thick circular head sitting into the hinge, or sitting into this pocket, basically creating a hinge that can only move up or down. Right? Your elbow joints, you can only really move up or down. You can't make it bend sideways. If you do, you're really moving your shoulder instead. Right? These two basically stay in planes with each other, or in the same plane with each other. Our pivot joints, such as between the atlas and axis, and then our proximal radial ulnar. But if you can just remember that one, you're great. It just allows for your neck to rotate without your entire spinal column or vertebral column needing to move as well. So giving that rotation in just the one plane again. Okay. This movement is moving your vertebral column. So if you go on a different plane, your vertebral column is stretching. Okay. So we have two biaxial examples here. Our ellipsoidal and saddle joints. We talked a good bit about the saddle joint with our discussion of opposition with that first carpo-metacarpal joints. 3 and 4 are also technically modified versions of this saddle joint, but if you remember that the saddle joint of the thumb is what allows it to not only go like that, but also go across your palm, providing for that opposition. And then we have our ellipsoidal joint, where the radius meets our carpals. A lot of our metacarpophalangeal joints, just kind of a little pocket that allows for that same biaxial movement. And then lastly, the ball and socket, which provides the most mobility. We just have our two examples, the shoulder and the hip, but they're also the least stable or the easiest to injure because of the structures that allow for that mobility. Right? We just have this little ball sitting in its socket, and we can't have too many ligaments and other connective tissue in there holding it in place, or else that wouldn't allow for the type of movement that we need those joints to have. So because there is less of that dense connective tissue, they're also the easiest to damage. Then lastly, our intervertebral discs. I think for this, like I don't intend to really test a whole lot, and maybe that is an oversight by me. Maybe this is more important and I should do more on this. But really, if you just recognize that this is essentially a squishy little pad in between each of your vertebrae, which tend to break down and get thinner and thinner over time as you age, which is what leads to like the idea of getting shorter as you get older, right? And shortening. It's because of these little pads and losing the fluid that's supposed to be filling these pads. It's just a gelatinous shock absorbing pad that sits in between each of your vertebrae to help resist that force, give you a little bit of flexibility with your vertebrae, allow it to bend and do the movements that your body needs from that, while also protecting it. Two main structures is our pulp, our pulposus, or the nucleus, and then the annulus fibrosus, which, as the name suggests, the annulus fibrosus is very fibrous. And then the nucleus, I guess this is a herniated disc where that has shifted out of where it's supposed to be. But the nucleus pulposus is more of that fluid and fat, and the blood vessels that supply it. So yeah, again, a gelatinous, squishy little pad that sits in between your vertebrae to protect them, allow for some shock absorption. So are there any specific questions I can answer about any of those topics from articulations?
Just quick questions on articulation. Do we need to know like the movement, but do we need to like the fibers, the cartilages, or bony fusion for each?
This stuff, is this what you're referring to? The part of this category? I think this is what you're referring to, right? I guess I think the only fair way I can say is like yes, that information is fair game. These names, the category is more likely to show up than the type. But knowing the type does play into the physiology aspects, right? The function of it. So generally being able to recognize these categories, a suture, gomphosis, synchondrosis, synaptosis, syndesmosis, symphysis, and then our synovial definitely could come up. And then recognizing their type of connection at those articulations is going to be helpful in recognizing their general function, and thus like their type of movement or if they move at all, etc. Don't know if that really helped at all, besides saying like, yes, it's fair game, essentially.
Okay, thank you.
Anything else here, you guys? All right, let's move on to muscle. So yeah, basically the topics you guys identified were like the same ones that we spent most of the time on Thursday, which makes sense. This is the most complex part, right? All the steps and stages, and all the little details of our myofibrils. So the general structure of muscle is bundles of fibers within bundles of fibers within bundles of fibers. And the muscle itself is just one entire organ, which would be each individual muscle has its own epimysium. Within that epimysium, we have little bundles like what you see here and what you see here called fascicles. And those fascicles are going to have, or this is one fascicle, sorry. All of these bundles are individual fascicles, and this is one fascicle. Each fascicle has its own connective tissue surrounding it. That is the paramecium. And again, these are the connective tissue is the really tough part of meat. Like the reason, or a majority of the reason that we need to cook meat in order to be able to chew it is because we need to break down that tough connective tissue from the epimysium, the paramecium, and then the endomysium, which surrounds each individual muscle fiber. And then even within one muscle fiber, which is one single cell, we have myofibrils, which are what contain our contractile unit. So this is one muscle cell kind of cut open, right? We have the membrane of that cell, our sarcolemma. We have the extensions of that membrane, our T-tubules. We have our sarcoplasmic reticulum, which is what holds all the calcium to allow for our muscles to contract. And then we have our actual myofibrils, which contain the contractile units, the protein fibers that are going to pull on each other in order to actually shorten the length of our muscle. And so one single contractile unit is called a sarcomere. So myofibrils have a bunch of sarcomeres end on end, and what you can see better here. This is one sarcomere, there'd be another sarcomere starting here and another sarcomere running the entire length of that muscle cell. And each sarcomere shares the same basic structure. Again, I don't assess on Z line, I band, A band, H zone, M line. I guess out of all of these, you should recognize M lines because it's the middle. It's where the thick filaments are going to be pulling the thin filaments towards. And that M line creates the middle for the contraction. But you won't see these other names show up on my assessment. And in general, these sarcomeres are the thick bands, which are myosin, pulling on the thin bands, which are actin. And so those myosin heads, we have them pointing in opposite directions on either side of this M line, and they will perform that power stroke, which will attach to the thin filament and pull them towards the M line, towards the center. And thus, this is happening in all of our sarcomeres, or all the sarcomeres that are innervated by the same neuron, which is going to cause that entire cell to contract, to shrink in size. And so we have multiple types of protein making up those sarcomeres. The thin filament has 5 different proteins. The thick filament really just has 2, myosin and titin. The ones to pay attention to are troponin, tropomyosin, and then recognizing G-actin. So G-actin, G standing for globular, think of like a glob. And so that literal glob-looking protein here, this little ball, that is the G-actin. This is what is going to attach with the myosin. They have these active sites, which are able to bond with myosin. But those active sites in a resting muscle are covered by this rope-like tropomyosin. And so that's the green looking rope there. It is covering the active sites of our G-actin. And then troponin is like the security guard holding the tropomyosin over those active sites. And so troponin is also going to be responsible for moving tropomyosin off of those active sites when it's time for our muscles to contract. Thick band or thick filaments really just recognize myosin. Titin is just kind of like the anchor. It's this little springy protein that's anchoring thick filaments and thin filaments to the Z line. But really just myosin is the main protein we're looking at with our thick filament. And so the myosin head is going to be responsible for attaching to these active sites on our G-actin, and then it will pull, do this power stroke that you see outlined there, which pulls all the thin filaments towards the center, towards that M line. Any questions of clarification about that before I move on to our step-by-step process of how our neurons lead, or how a nerve signal is going to lead to the contraction of the muscle? You're muted, Constance.
Sorry, so the titans will expand and contract depending on the length of the sarcomere, right?
Yes. Yeah, right. They have that spring-like structure. They're flexible, so they can shorten and extend, which plays into our muscle's ability to extend up to 3 times its length. And yeah.
And so the sarcolemma of the muscle cell would be surrounded by the endomysium. Would that
Yes.
correct?
Yes, there would be endomysium connective tissue surrounding the entire sarcolemma of each muscle cell.
Thank you.
Okay, so muscle control. Okay, so we talk a little bit about the axon, or sorry, the neuron that is going to send the signal to our muscle, and then we talk a lot about how the muscle responds to that signal. So basically, we have neurons that are stimulating one or a few or a lot of muscle fibers all on their own. Where those neurons meet the sarcolemma of the muscle is called the neuromuscular junction. And so the neurotransmitters that are going to be released from that neuron end up creating a response on the membrane of the muscle cell, which will eventually lead to those contractile units, the sarcomeres, contracting. Okay, so we're going to talk about all the steps from the action potential of the nerve all the way down to the actual muscle fiber itself contracting. And it's all kind of summarized here, but we'll look at the step-by-steps of it. So this is all surrounding the idea of action potentials or membrane potential in general, essentially at rest, which is kind of something we're going to talk about today. Let me actually pull up a slide from that. It's from today's chapter. Is it here? Right. So at rest, our neurons and muscle cells are maintaining a membrane potential of -70 millivolts, which is measuring the general charge inside the cell compared to outside the cell. So there is a negative charge on the inner portion of the membrane inside the cell compared to the outside of the cell. The outside is more positively charged. And this potential of -70 millivolts is constantly maintained. Okay? So at rest, there is a higher concentration of sodium and chlorine outside of the cell and a higher concentration of potassium, proteins, and phosphate inside of the cell, which all play a role in this charge. And we have some protein channels that are constantly open to allow for diffusion of each of those ions. And then the combination of concentration and charge produces a electrochemical gradient, which drives where those ions are moving. Okay? So without getting into too much of that detail, by changing the concentration, by forcing ions to move in one direction or another, we are going to basically activate some other proteins that are gated either by a chemical, and they will open when they bond with that chemical, like acetylcholine, or gated by voltage. So by forcing the movement of ions to one side of the membrane or the other, we change the membrane potential, which is going to open different protein or voltage-gated protein channels, and thus allow for more movement or more directed movement, which is going to cause a domino effect of that same change in membrane potential to continue to disperse along the membrane of that cell. And so in the example we're about to look at with how neurons stimulate muscles, an action potential moving down the axon of the neuron through these voltage-gated channels. Okay? So this change in membrane potential here is going to cause that domino effect all the way from the body of the neuron all the way down the axon until it reaches the neuromuscular junction. When it gets to those terminal knobs in the neuromuscular junction, that's going to open other voltage-gated channels that are going to cause calcium to rush into the neuron. Oh, I guess we didn't really talk about that portion. Okay, cool. We don't need to. Basically, the action potential from the nerve, from the neuron, gets all the way to the terminal knob. So where the neuron is meeting the muscle. That action potential is going to stimulate calcium to enter, which is going to cause these vesicles to release acetylcholine, a neurotransmitter. That's what we see here. The action potential has reached the terminal knob. So now these vesicles are releasing the acetylcholine into this synaptic cleft. That acetylcholine is going to bond with some of those ligand-gated channels. These chemically regulated channels here. So here's an acetylcholine channel. Acetylcholine binds with the protein changes shape, opening up, and now a bunch of sodium is going to rush into the cell, causing another action potential. This is basically transferring the action potential that was traveling down the neuron to start an action potential that is going to travel down the muscle cell. So acetylcholine opens those protein channels, allowing sodium to flood in. That's going to depolarize the potential of the membrane, causing another action potential. So now we have a domino effect action potential that is going to travel down the entire membrane of the muscle cell, so down the sarcolemma. And so as that potential leaves, and that domino effect is continuing to go down the sarcolemma. Our sodium potassium pump is going to return our membrane potential back to rest. Acetylcholine esterase, this ACHE, is going to clean up that acetylcholine and give it back to the neuron. And so that action potential has ended. That signal has been transducted, and now we're back to where we were before that action potential ever arrived. Okay, so that is just the beginning of how this started. This is just transmitting the action potential that was on the neuron to now be traveling on the sarcolemma of the muscle. So if we follow what that action potential is going to cause, I guess we need to go back a little bit to get the best show of this. So the neuron caused an action potential to start on our sarcolemma, the membrane here. That action potential is going to travel all the way along this membrane. It's going to travel down into our T tubules, which are just extensions of the sarcolemma, into the cytoplasm, the sarcoplasm of the muscle cell. That action potential is going to cause the opening of some other channels, essentially releasing calcium from our sarcoplasmic reticulum. So the action potential from the neuron caused the release of acetylcholine. Acetylcholine caused an action potential to start on the sarcolemma. The action potential on the sarcolemma went down the T tubules to stimulate the release of calcium from the sarcoplasmic reticulum. Right? The T tubules always come in between two different sarcoplasmic reticulums, and each of our sarcoplasmic reticuli have these terminal cisterns full of calcium. These are just calcium pockets waiting to be released, and when an action potential gets there, they release the calcium. So again, action potential from the neuron released acetylcholine. Acetylcholine started an action potential on the sarcolemma. The action potential from the sarcolemma released calcium from these terminal cisterns. And now that that calcium has finally been released, the muscle is now able to contract. Because that bodyguard we were talking about earlier, troponin, it will only move when it has calcium to bond with. And so now that calcium is leaving our sarcoplasmic reticulum, there's a bunch of calcium available to bond with troponin. Troponin changes shape and moves that rope-like tropomyosin away. And now myosin can bond with our G-actin and power stroke. So we'll look at that step by step, right? So at rest, tropomyosin is covering all of the active sites and troponin is holding it there. When our calcium is released, it will bond with troponin, causing troponin to change shape, which is going to move tropomyosin. And now these active sites are available. The myosin head is going to bond with those active sites, and it's been waiting for that the entire time. As soon as that becomes available, myosin is immediately going to try to attach. It will release this ADP and free phosphate molecule to cause it to change shape again, which is the power stroke. Right, so it's now gone from this extended formation to a flexed formation, pulling that thin filament towards the center. And now, in order for it to let go, or basically that is contraction. So calcium allowed troponin to move tropomyosin. Myosin bonded with the active site of the G-actin, and then released the ADP and phosphate to do that power stroke. That is contraction. So now to allow this muscle to relax, we need to disconnect this myosin head from that globular actin. And so adding a new ATP molecule will allow that myosin head to release the G-actin. And if this calcium starts to be removed, which basically just comes from a concentration difference, to connect it to Constance's question, which I'll go back to in a second. So ATP allowed the myosin head to let go. Calcium is going to start being cleaned up and move those tropomyosin strands back over the active sites. And then the hydrolysis of ATP into ADP and phosphate is what puts the myosin head back to where it started. So in some cases, that calcium isn't going to be removed right away, and that's going to allow for repeated contractions very quickly as we continue to send that signal through our nerves, telling our muscle we want it to continue to contract. But if those signals stop, then there's no more action potential causing those channels, those calcium channels, to stay open. So the sarcoplasmic reticulum will basically close shop. It's no longer releasing calcium. And then the diffusion, the concentration difference basically is going to push calcium to go back to where it was. Or essentially, it's the charge difference, the combination of concentration and the electrical gradient. So the entire electrochemical gradient basically pushes calcium back to the sarcoplasmic reticulum. And that's how it gets released from troponin and gets back to where it was. So lots of steps summed up pretty quickly in this figure here, which is nice. And then the end of muscle contraction also summed up here. These are probably your best friends as far as trying to quickly review this stuff and get a larger picture. The videos, any kind of process that we're learning about, watching videos that have animations are going to be the best way to learn that. So please watch those suggested videos on Canvas if that is hard to follow, which I definitely recognize it is. It's a lot of steps. So again, I shared this before, but thinking about how many steps it takes just to contract one sarcomere and thinking that there's thousands to millions of sarcomeres involved in a movement like this, and that's all happening in an instant to us, but all of that needs to happen at that small of a level, like the relativity of time just seems crazy. So many things happening in what we consider an instant. Great. Are there any specific things I can clarify about that? Okay. So I know we don't have a whole lot of time, but some of what we were just talking about with neuromuscular junctions and muscle contraction control does overlap with kind of what we go into more detail with in chapter 12. And so it might be helpful to talk about, right? Expand on this basically. So just a brief introduction to some of these slides, and we'll go over this in more depth on Thursday. But all of our neurons have some of the same basic structures. There's a body, which is what holds the majority of the organelles. There's a dendrite, which is what is typically receiving a signal from something else, and then an axon, which is going to carry that signal to the next neuron or the target cell. Okay, so with what we were just looking at, we were just looking at the last neuron in a chain of neurons, which is the one that meets the muscle cell. And so they meet with these little branches at the end of our axon called telodendria. And at the end of each of these branches, we have these terminals, which are basically just holding whatever signal that neuron is going to send out to the next cell. So in the case of muscle cells, we have a bunch of vesicles holding acetylcholine that are just waiting for the right signal, waiting for an action potential to get here, telling them to be released into this space, and then causing another signal on that muscle cell. And so a lot of the time, and in the case of the neuromuscular junction with muscle contraction, that neurotransmitter that's being held in that knob is going to activate a chemically regulated channel. So we release the neurotransmitter, that neurotransmitter bonds with a protein, a membrane protein on the target cell, and that causes that protein to change shape, which is typically going to open a channel like this one, which allows sodium to rush into that cell. Okay. And basically the rushing in of sodium into any cell is what is going to cause most action potentials. Basically every example of exciting a electrically signaled membrane is going to come from sodium rushing into the cell. Okay? It is typically in higher concentrations outside. It is an extracellular ion. And so when a channel opens, allowing it to rush in, that's going to be a big change in the overall electrical charge of that space, which is going to cause a chain reaction from these voltage-gated channels. Okay, so basically what we see here, if we combine these, so acetylcholine allowed for sodium to rush into the cell. Sodium is a cation, it carries a positive charge, so that's going to increase this number from -70 up to -60. At -60, more sodium channels open. So this time, voltage-regulated channels are going to open, and so even more sodium rushes in. So that number is going to start increasing at a much faster rate, up to around +30 millivolts. And typically at plus 30, these channels are going to start closing. The sodium channels are going to close, and other, like the sodium potassium pump, is going to get our membrane potential back to normal. But because this depolarization has happened, we've taken this negative number and made it more positive, and that has caused more and more of that. These ions are going to be basically continuously pushed like a wave down, causing that domino effect of more and more of these channels to open and open and open as it moves down the membrane of whatever target cell it is on. And so that's what we were seeing with that neuromuscular junction. Okay, the nerve had an action potential of these channels opening and opening and opening to cause this action potential to travel down the axon. So it started at the axon hillock, and those channels opened like a domino effect all the way down the axon until they reached our synaptic terminals. At that synaptic terminal, that allowed for the release of acetylcholine from that into the space. Acetylcholine bonds with these proteins on the muscle, opening this sodium channel. So as sodium is rushing in, it's going to depolarize the membrane a bit. It's going to take that -70 number and push it up towards -60, which opens up more sodium channels. And so more sodium rushes in, and that same change is going to keep occurring like a domino effect down the sarcolemma. So the action potential continues until it gets down the T-tubules, and is going to open more voltage regulated channels, but in this case, calcium channels from the sarcoplasmic reticulum, so that now calcium is being released into the sarcoplasm. Okay. Trying to see if there's anything else that helps there. And that's it. That's all I wanted to say here. Hopefully that helps a little bit with that making sense. And yeah. So that's where I'm going to end today, unless you guys have more questions or concerns, things for me to talk about. But hopefully that was helpful in wrapping some things up, getting some of those confidence numbers up as you prepare for the exam tomorrow in lab.
So, sir, the release of calcium from the sarcoplasmic reticulum is initiated by changes in voltage
Yes.
voltage channels.
Yep.
All right, thank you.
Yes, it is a voltage-gated calcium channel. All right, guys. Thank you for your participation with this. I hope you have a great rest of your day, and I will see you tomorrow for our Unit 2 lecture exam.
Thank you.